Welcome to ALS Today. This is Michelle Flom reporting. Today, we turn our attention to hyperexcitability, the overactivity of key neurons in the brain and spinal cord, a potential early step in ALS. Joining us to talk about hyperexcitability is neuroscientist Steve Vucic of Neuroscience Research Australia in Sydney. Thank you for joining us today, Steve.
Thank you, Michelle. It's a pleasure.
What is hyperexcitability?
Well, hyperexcitability is a process within the brain where there is excessive activity of a particular molecule called glutamate. People normally use glutamate for various functions like learning and communication and so on. What happens in motor neuron disease or ALS is that there is too much excitotoxicity of glutamate activity, which is harmful.
What made you first suspect that hyperexcitability could be an early step in ALS?
Well, the role of corticomotor neuronal hyperexcitability really stems back to Charcot, who described the disease and coined the term amyotrophic lateral sclerosis to describe what he was seeing both clinically and pathologically, whereby he found that in addition to sort of lower motor neurons dying, there were these upper motor neurons that were also not quite right. And he initially proposed that perhaps it's these upper motor neurons that are causing downstream effects. And subsequently, a colleague of ours, Andrew Eisen, proposed this dying forward hypothesis, where he proposed that motor neuron disease or ALS begins centrally, and it's these corticomotor neurons that mediate anterior horn cell death through an excitotoxic process. And it's really those two theories that have driven our research to determine whether in fact they were correct or not. Because determining whether cortical hyperexcitability is a process is important from a therapeutic perspective, because it would tell us where motor neuron disease begins, what the process is, and where one should direct one's treatment, whether it's at the spinal cord or at higher levels.
So you took a look at people with ALS using certain techniques. Can you tell us a little bit more about that?
The technique that we use is called transcranial magnetic stimulation, which allows us to stimulate the brain and measure the level of excitability. So we looked at a cohort of sporadic ALS patients and found that hyperexcitability was evident in this group, and it was an early feature and linked to peripheral neurodegeneration. We then looked at a group of mimic disorders, that is, diseases that look like motor neuron disease but are not motor neuron disease, conditions such as Kennedy's disease and various autoimmune neuropathies, and we found that their level of excitability was normal, comparable to healthy controls. And so that suggested to us that hyperexcitability is not a simple plastic process, but rather something intrinsic to ALS. We next looked at a cohort of familial ALS patients who had the SOD1 mutation, and we found that in the clinically affected patients, they had hyperexcitability. Again, also, as with sporadic ALS patients, this hyperexcitability was linked to neurodegeneration. However, when we looked at the carriers, most of the carriers were normal. In a small group of these carriers, we noticed there was an excessive level of excitability, and on follow-up, we determined that unfortunately these patients went on to develop clinical ALS within, on average, 5 months of our testing. So that to us suggested that hyperexcitability is intrinsic to both sporadic and the familial SOD1 ALS patients, and that it's probably an initial event that drives the ensuing neurodegeneration. And more recently, we looked at the C9orf patients. The C9orf is a gene that was recently described. It's probably the commonest genetic mutation in ALS, and lo and behold, we also found that this cohort of patients were hyperexcitable. And again, this was linked to peripheral neurodegeneration. We are currently looking at the carriers of the C9orf gene and following them up to determine whether hyperexcitability will also, in this group, precede the development of the syndrome, that is, the development of the peripheral weakness.
So what I think you're saying is that not only can you detect in patients the sign early in the course of their disease. But you're also saying that you don't see this in carers, which suggests to me that this may be something that clinicians could use to start identifying people even before they show signs of disease.
Correct. I mean, one of the other interesting aspects about this test is that it could be used as a diagnostic procedure. We looked at a large cohort of our sporadic ALS patients to determine the diagnostic utility of this test, and we found that we could make a diagnosis of ALS 8 months sooner with this test than if we didn't use this test. And that could be potentially important because it's those 8 months that could be the critical therapeutic window for institution of neuroprotective therapies. On the same token, it could also mean an earlier recruitment into treatment trials, and perhaps that could lead to successful therapeutic trials. At this point, we are recruiting patients relatively late into these treatment trials because of the diagnostic dilemmas that we're faced with. We don't have a diagnostic test for ALS, and as such, we have to rely on various clinical criteria and also exclusionary tests. So with the cortical excitability testing, we think that this could identify patients at an earlier stage in the disease process, and perhaps they could be more amenable to therapy at that stage.
So based on your initial results, it sounds to me like it has a lot of promise, not only in terms of timing, but also being able to rule out other diseases that look so similar.
Absolutely. This test reliably distinguishes ALS from mimic disorders.
What else do you think really needs to happen to be able to vet these techniques to get them into the doctor's office?
Well, I think an important component of that is commercialization with an industry partner that can support such technology. We are in the process of doing that through a UK company called Magstim, which is the largest producer of these transcranial magnetic stimulation devices. Once the hardware and the software is all geared up and wired up, then I think this test could potentially be useful. So I would hope that over the next 12 to 24 months, we will have this test clinically available to the ALS physicians throughout the world.
Which is really exciting to see that it's finally coming into play. That must be very rewarding. So one of the other things that I'm thinking about is what I started with, that we could begin to get people into trials sooner, but at the same time, it's also suggesting new ideas to treat disease. So to sort of get started on that topic, what do we know exactly about hyperexcitability and the underlying mechanisms? What causes it? Why does it happen in people with ALS?
The precise mechanisms underlying this are still not fully worked out, but there are potentially a number of interacting mechanisms. One of the cells that are quite important in controlling the level of brain excitability is the astrocyte. The astrocyte mops up the glutamate once it's released from the presynaptic nerve terminals. Now, we know that in ALS patients, the astrocytes are dysfunctional. They're not working, they're not mopping up the glutamate. And that's one source of glutamate excitotoxicity. On the other hand is the lack of inhibition. So normal cortex has a level of inhibition which is mediated by these inhibitory neurotransmitters. And what we know in ALS, these neurons that sort of work by the GABA transmitters, the so-called inhibitory cortical neurons, degenerate. So there are multiple interacting mechanisms that are mediating glutamate excitotoxicity. And it's exciting because we're now getting to a cellular level, that is cellular targets that can be potentially modulated through either stem cell techniques or other techniques that could potentially lead to a better treatment for motor neuron disease. One of my hopes is that we can use our technique to detect the disease very early when the patients are minimally affected, and then we could sort of arrest this process by targeted stem cell therapy, and so the disease does not spread. So that's the hope for the future.
So one thing that I'm thinking about right now is the work of Michael Benatar's group in the University of Miami, and he is now collaborating with Martin Turner at the University of Oxford to start to be able to use certain imaging techniques and other techniques to be able to identify people who are at high risk of ALS to developing ALS. So sort of thinking about all of that, are you also thinking about working in that area to try and find people earlier?
Absolutely. Martin Turner and Michael Benatar's work is very important, and they're doing some wonderful work on imaging. We've started to look at various novel MRI techniques that measure the anatomical structures within the cortex, and we're finding very similar results. And certainly their results are reinforcing what we found, that the upper motor neurons, or the central nervous system, seems to be important in motor neuron disease. This notion of disconnection of various central pathways, leading lack of inhibition, excessive excitability of these various networks, that's all sort of pointing to one direction, and that is that the upper motor neurons, or the central nervous system, seems to be quite important in ALS pathogenesis. And again, from a pathophysiological perspective, it just underlines this key notion that the brain is very important in motor neuron disease. And that is further underscored by this C9orf discovery, which suggests to us that there is a continuum between ALS and frontotemporal dementia. It's all sort of within the same rubric. It's quite nice for Charcot and Andrew Eisen. They were right all those years ago.
What I'm hearing is we're now finally being able to really trace back this disease into the brain. We have techniques that can find this disease even earlier than we have been able to do in the clinic, including this transmagnetic stimulation technique that you're talking about. And we're going to probably in the next few years hear a lot more about being able to find these patients sooner, diagnose them sooner. But at the same time, we of course also need better treatments. And I think one of the things that's really exciting about your work is it's giving us some clues as to what could be going wrong way early in ALS. So one thing that I was also thinking about was Bob Brown has recently, I think it's not quite published yet, suggesting that there are certain drugs, such as this FDA-approved drug, mixilotine, that may reduce hyperexcitability and therefore be an earlier treatment for the disease. I know that he has shown in mice that there is a survival benefit, and this is soon going to be going into clinic this summer. It's a trial that's led by Mike Weiss from the University of Washington. So I'm wondering if you could share your thoughts on that and other small molecule strategies that maybe could help us treat it.
Absolutely. The work with mixilotine is very exciting. Mixilotine is an agent that would reduce the level of nerve excitability, and it's got a potential to work. What's important now is for us to see whether it's going to work in humans. There's a lot of drugs that have worked in mice, but unfortunately not in humans. However, mixilotine is a sort of different type of drug to what has been used in the past. It's like riluzole. It works by dampening down the level of nerve hyperexcitability. So one would hope that with a combination of anti-excitotoxic agents, we could finally get a good treatment for this devastating disease. And there are a lot of other neuroprotective agents that work in a similar light. For example, one of the trials that has also been touted, albeit at a preliminary stage, is gilenial fingolimide, the MS drug, which works in part by acting on these non-neuronal supportive cells within the central nervous system, such as the astrocyte. So the future is, I think, very exciting, but we have to get a move on because our patients, once afflicted by this disease, really don't have a long time. So I'm very excited by Professor Brown's work, and I'm looking forward to the human trials for mixilotine.
Absolutely. So Steve, I want to thank you for joining us today and telling us about your work.
Thank you. Take care.